called Pangolin to aid in those efforts. And so instead of butchering it by talking about it myself, I think I'm just going to pass it over to Aine, and she can go over what the tool does and kind of give us an introduction. So I think, Sean, if you want to start recording.
Yeah, already started.
All right. Oh, perfect. All right. So Aine, I'm going to pass it over to you.
All right. I'm going to try and do this with presenter tools this time, actually. So I'll just, if I go into slideshow and then presenter view, I think that should, does that look normal to you guys?
It's in the presenter view, I think. So we have two slides next to each other.
Oh, that's okay. I can not do that. And just do a regular one. Okay. It all goes so smoothly in the trial run, doesn't it? Okay. Okay. So yeah, I'm gonna, like Logan said, I'm gonna talk about tools for using genomic epidemiology to inform public health, which is basically what we're all doing, really, isn't it? Okay. Yeah, firstly, I wanted to say thank you for inviting me to speak. I'm excited to share with you what I've been working on over the last while. I guess the structure of what I'm going to talk about today, initially I'll talk about lineages, why we're using lineages to try and track the genomes of coronavirus. And then I'm going to talk about how we're assigning those genomes, and then also the sort of more practical applications of how we've been investigating outbreaks on a daily basis. I don't need to tell you guys the huge spread of coronavirus that's gone across the globe this year. We've all been impacted by it and are working flat out on it. But as well as that, there's been this incredible genomics effort, which as of yesterday, when I looked this up, there's 73,000 submissions to GISAID. And with so much time and effort invested in SARS-CoV-2 genomics, it's crucial for this information to be fed back as soon as possible and to try and get useful information out of this data. So how do you get useful information out of this many sequences? I guess the first thing to do is to define clusters so you can tell different clusters apart and tell apart different local outbreaks. We want to be able to identify things like imports and exports to countries or to more local levels. Ways you could break up the tree. These are three systems that we've been looking at. Phylotypes are a sort of very fine scale information, and we define them by codifying up the tree, so they've been really useful for us to actually tell apart closely linked sequences or to rule out transmission. They're not stable assignments, but when we've been doing outbreak investigations, we assign phylotypes, and we found this quite a useful label to feed back into public health. Clades are sort of the other end of the spectrum. They're more broad stroke groups based on SNPs across the phylogeny. Really useful for tracking trends over time. Systems include NextStrain and GISAID, and this figure I created with Emma Hodcroft together when we were trying to compare different clade systems versus the lineage system. Lineages are sort of finer scale, and they're designed to try and capture the edge of the epidemic. They're more suited, I think, to outbreak investigations and to tracking on finer scales. As you can see in the figure, it's a hierarchical system, and in addition to the structure of the tree and the genetics, we also use epidemiological data. So it goes beyond just splitting up the tree into chunks. We define it as an introduction into a distinct geographical area, And then we look for evidence of ongoing transmission, which sort of, when you look at the phylogeny, it translates into either internal nodes within that part of the tree. This is an example of a classic lineage. So in the background, you can see sort of, these are European sequences in the background. And then there was a single introduction event, we think, into India. And then we see sort of evidence of local spread. So a distinct location. And as a general rule, we try and look for good support within the tree. So whether that's bootstrap likelihood or whatever measure you have. But we say this with a little bit of leeway because as you'll see, the tree is quite changeable. And an important thing I think to note is that the size of the lineage isn't relevant. You can have some incredibly large lineages, which we do, and some really small ones. So we know that the number of sequences doesn't necessarily reflect the number of cases, but equally, we're looking at spread within an area. If there's, say, this relatively small lineage in India, that is as useful, I think, a tool as the much bigger ones as well. So, just to give you a broad overview of the tree, if you guys have been working on SARS-CoV-2, you're probably very familiar with this, but A lies at the root of this global phylogeny that we've got. It's sort of the beginnings of the outbreak. We root on an A lineage from early in the outbreak that was taken from Wuhan. And here we see a lot of the diversity in China, but then also with exports across the globe. B is also, the base of that also lies in China with exports across the globe as well. But we have some independent individual B lineages, for instance, B.4, that is quite a nice example of using epidemiological data to assign lineages. So B.4 roughly equates to the large Iranian outbreak that happened earlier on this year, or that started earlier on this year. And we identified B.4 as the Iranian lineage weeks before an Iranian sequence or a genome came up online, purely by using metadata associated with sequences from around the globe. We had travel histories. B.1 is what is the sort of dominant lineage circulating around the globe at the moment. And you can see this long branch that leads up to it, but actually it's not that long branch that defines the lineage, so this equates to the long branch. In this section of the tree where there's the Bavarian outbreak, that includes a number of Chinese sequences from Shanghai and from Beijing, and actually because there's diversity within China, we don't define this section of the tree as part of B.1. It's the smaller branch leading into it that equates to the northern Italian outbreak earlier on this year. That was where we had this introduction and then sort of rapid amplification of cases. And within this lineage, we don't see any Chinese sequences that have been, that are at the root of this sequence, of this lineage. So having described the tree and how we're all on board now with how useful tools lineages are, I'm going to just discuss how we've gone about assigning lineages. And there are various ways of doing this, and I think people who work in bacterial genomics are used to using things like SNPs and types, and I think it's more commonplace in virology to use whole genome alignments because they're small. And really in this outbreak, it's sort of a question of this particular virus and what's the most suited to tool to use. And SNPs are really quick, but looking across the global phylogeny, we see a number of things that could result in misassignments. things like homoplasies, lab artifacts, and missing data are rampant across the tree. For instance, D614G, which is a mutation that people have been talking about recently. We've been calling it Doug, and Douglas is the ancestral form. But Doug is an interesting SNP, we think, because there's some evidence that it increases ability to transmit. But around Doug, we see that this is actually not a single mutation event. So all of the orange ones are D614G. But we see this has arisen a number of times across the tree. The grey tips here are a separate mutation, also at the same site. But we also see reversions. So when you're building a tree and you're assigning based on SNPs, something like this homoplasie here could really impact what assignment you get. And also, so this is just one example. There's been number of estimates about the amount of homoplasic sites across the alignment, and I've seen some estimates up to 30%, maybe lower. Some could be due to lab artifacts, but definitely things to keep in mind. Depending on protocols that people have used, I know the Arctic protocol has been really popular, but there have been cases of Amplicon dropouts, and this missing data actually at the Doug site is one of the common missing Amplicon, Amplicons. That can also lead to misassignments. So we have adopted the approach of using trees from full viral genomes, and with the hope that using the context of all of the diversity, that this may account for things like missing data and stuff like that. So of course, building trees is heuristic, and especially with this many tips, like we're coming up on 70,000 tips in a tree at the moment, the tree space is enormous. So you can be searching and think you have a good tree, but actually you're stuck at some local maximum, and the tree is over the other side of tree space, and you haven't searched it. So the thing is, we know this, but we also think that it's probably more useful to be still using trees and to try and incorporate all of that diversity that we can. So Pangolin 1.0, I don't know if you have been on Twitter, but we recently released Pangolin 2.0. But 1.0 has done us very well up till now, and it works from an anonymized alignment and a guide tree, and both have representatives from each lineage that has been defined. The lineages are assigned manually by curating the global tree, and there's a prep pipeline that assigns and selects the different representative sequences, trying to maximize the representation of diversity within each lineage and also minimize N content within each lineage. And we mask out terminal ends, and we mask out singleton SNPs as well, to try and account for sequencing error, and which particularly at the ends of the alignments, you guys will probably know that that's more common. And very simply, you take your input sequence, your input whole genome, and you're not sure where it goes. We stick it into the guide tree, and using an algorithm that J.T. Crone wrote, we assign the most likely lineage, and with that we get a likelihood value and a bootstrap value, and really straightforward. It just spits out a CSV at the end. If your sequences fail, there's some mild, I'm going to say mild QC that your sequence will go through, but it will tell you why we didn't assign it if your genome doesn't get assigned a lineage. One of the big developments that's really helped make Pangolin accessible to people globally has been that we have an interface, and this was developed by a team led by Anthony Underwood at the Centre for Genomic Pathogen Surveillance, and we've sort of affectionately called it pangoLEARN, but the official name is the web application. So this has been very simple. It's a drag and drop interface, and you're able to get the same information out, summarizes some of the information about what sequences are out there per lineage, and then it links out to microReact, which is a visualization tool that the Centre for Genomic Pathogen Surveillance has developed. So you get an interface like this. You can see what the lineage looks like around the world, where your sequence is, how many samples have been there over time. And also microReact has interfaces like this that summarize lineages across the globe. So it's been really useful to be able to link up with that, and particularly when trying to bring genomics to the clinical side of things, if there aren't necessarily people who have bioinformatics experience, it's really useful to be able to point them towards this as well. So I mentioned that we have Pangolin 2.0 released now, but up until we released that last week, on the interface alone, there were 150,000 unique sequences assigned by Pangolin, which is really amazing to think about the amount of genomics work that's been going on globally. So each data release has provided an updated guide tree, an updated alignment. Back in April, when we did the first official data release, we had 261 tips on the tree, and each lineage was represented by a minimum of 5 sequences. A couple of weeks later, the tree had almost doubled in size, and then for the next data release, we reduced it to 3 representatives. But as phylogenetics doesn't scale particularly well with increasing tips on the trees, Pangolin was getting a lot slower. So a data release I started working on in June, we, even with reducing to just 2 representatives per lineage, and think about something like B.1, which has thousands of sequences in it, picking 2 representatives from that was quite a loss of information. And with that, the tree was still getting bigger and it just wasn't scaling. So we had to upgrade, and we've released Pangolin 2.0, aka pangoLEARN, which is developed by Emily Scher. And this is our first instance, our first attempt, and it's very simply just a multinomial logistic regression. And the model was trained on 30,000 sequences using scikit-learn, and then the manual lineage assignments. So I still went through the tree manually and assigned lineages. And I think there was about like the 30,000 of them there, so it takes a while. Benefits of pangoLEARN are that it's faster, and equally, as the tree has gotten bigger, we're now taking all the diversity into account. So all of the sequences go into training Pangolin, rather than picking just a representative few sequences as we have been doing. So, and a benefit of this as well is that if you have homoplasies across the tree, they should just be uninformative. So this was the first release of pangoLEARN, and the next steps are to try and implement a hierarchical model. We actually were surprised at quite how well this simple multinomial regression actually did. We got really high accuracy, really good precision, but of course smaller lineages do worse because there's less data to train it. So we have been talking about the next release, and there are things we're going to be trying. So we've talked about lineages, assigning lineages, but actually how are we getting this information back to public health individuals and bodies? And also, I know I'm just one person. So I recognize that the updates in lineage releases aren't coming as quickly as they're needed. The tree is like 70,000 sequences big now. So what we're going to try and do is outsource and get more people involved with lineage assignments. So this is what llama is, local lineage and monophyly assessment. It's a new tool that we've been working on. And in the first instance, you can use this to check whether your sequences are monophyletic lineage, and that means that you can contribute to the lineage data releases. And the current system that we've got set up is via GitHub pull request. So really straightforward. What llama does is pulls out local trees from a large tree, and you can, at a minimum, give it a set of query sequences, query IDs that are already in the tree, but it can optionally add in new sequences to that tree on the fly as well. Dependencies, because obviously to pull out sequences from a big tree, you need a big tree. So you need a big tree. If your tips that you're interested in are already in it, then that's all you need. However, if you want to add in sequences on the fly, you'll also need an alignment, which at the moment would have to be gotten from GISAID, but we're talking about potentially hosting an interface that we can have the alignment just for data release issues and privacy issues and all that with GISAID. And then also just your set of query sequences. So quite a good resource, if you're not aware of it already, you might be, is Rob Lanfear's global maximum likelihood phylogeny, which he's updating regularly, which is well constructed. He's using FastTree to make it, also doing quite heavy masking of the ends of sequences, potential homoplasic sites. But for pulling out sequences, local trees with llama, this tree is already up and available, but of course if you've got your own set of infrastructure, you could use that as well. And then you can do bulk downloads of sequences on GISAID as well in the meantime. But for llama, the actual process that it goes through, in the very basic sense, if you have your input CSV of query tips that you want to pull out, it checks the global tree. If they're in the tree, it will pull them out and show you a summarized set of tips around your sequences of interest. So if you have a set of sequences, you can very easily tell whether or not it's a single monophyletic lineage or multiple lineages. If your sequences are present at opposite ends of the trees, it will pull out multiple trees. You can customize how big your local trees are that you pull out. So there's a distance metric, so you can count 1, 2, 3 nodes away from your sequence of interest. If it's not in the tree, if you supply an input FASTA file and your alignment, we then run some QC steps in terms of length and end content on the fly. We map and pad against a reference, we map against the alignment, and we find the closest sequence. So the mapping steps are done with Minimap, and the padding is done with DataFunk, which is custom software that we have as part of our lab here. All of this is open source as well, so you can check out the code. If you've got some in the tree, some not in the tree, we can combine it all and then pull out the local trees. If you've supplied an alignment, we can pull out representative sequences for each lineage that's present in your local trees. And then we do a summarizing of the polytomies in the tree. So I'll show you in the next slide an example of what that means. But we take those representative sequences, we gather all of the sequences, root with an outgroup, and we can add your sequences into these local trees. So rather than having to reconstruct the entire global phylogeny again, you can construct these local relevant trees on the fly. and then optionally generate a report. So this is depending on your goals. So the report could be used to provide evidence that you've found a new lineage and do a pull request on GitHub. But as I'll show in a minute, this report can be extended and actually used to feed back into public health information about lineages, the sequences that you have. So when I talk about summarizing polytomies, just to be clear, this is what I mean. So a local tree, here as an example, it's still pretty big. So even though it's not, 70,000 tips, it's still a few thousand tips, because I don't know if you've been looking at SARS-CoV-2 phylogenies, but there's a huge amount of identical sequences being sequenced, and we have these sort of shared polytomies, which is great if you're interested in them, but if you're, for instance, interested in these three sequences here, actually this diversity is not necessarily relevant to you. So what we do is we collapse on polytomies, and as part of llama, we find a representative sort of set of sequences per lineage. So imagine this is a red lineage and this is a purple lineage. We add in a few sequences just to sort of give you a bit of context. And we have all this information about what's in each node collapsed, so that you're not losing information, it's just summarizing it for you. And for example, I just pulled out a couple of example sequences from the global tree, and pretended that these were not in it by writing closest. So the report that gets produced, you get what the lineage is, which tree they're in, and then as part of the report, it also visualizes in a nice, pretty way, the trees, the local trees. But you also get the sort of tree files as well, so you can do what you like with those. Just as an example for the usefulness of this, a few weeks ago I was involved in a project working with Placide, where they had done a huge amount of sequencing in the DRC, and still are doing, but for them to try and get useful information out of the genomes, they've been putting up on GISAID actually, they've been really good with open data, but I manually ran some analysis and made some figures for them, and pulled out local trees by hand, and gave them some information about what lineages were circulating in the DRC. So with llama, the same analysis could have been done locally and quickly, so that when the genomes had been analyzed and the consensus sequences had been added into their database or whatever, they could just pull out that information straight away and generate a report within a time frame that's actually useful for public health. An extension of this, as part of our work with COG-UK, which is COVID Genomics UK consortium, we've been working on a tool called civet, which sort of a similar principle to llama, in that it pulls out local trees from this large global tree, but it's more specialized, and it's sort of, this is a UK specific tool, but it's showing you in theory what llama could actually be if it was developed to be more specific. At the moment, llama is a more general tool. So civet is based on the CLIME infrastructure, because we have this global tree being created every week as part of COG. And just to give you a bit of background, I'm not sure if you'll have heard of COG or not, but we are an academic-led consortium where we're gathering and analyzing sequence data for the public health response. There's over 12 universities and all 4 UK public health agencies are involved in the project. So every week we're reporting direct to the Chief Scientific Officer and also feeding information back to local public health services and scientists as well. So yeah, check out more information about COG here. The way it works is we have sort of local metadata and patient linkage occurring all across the UK. We've got local sequencing centres actually doing the physical sequencing. So some people have gotten gridirons for this project, some people are using MinIONs, some people have Illumina capacity already. And the bioinformatics is run locally as well. So we have people running maybe the Arctic network pipeline and things like that. So this has all been done locally. And then it started out once a week. It would be, all of the data would be uploaded to a central location, Climb, which is like a big server in the UK, and the sort of bioinformatics processing and some QC was done by Majora, which is a tool that Sam Nichols has made. The phylogenetics pipeline has been run in our lab in Edinburgh. We called it Grapevine. And Rachel and Ben have been taking the lead on that, and then Verity, who's also in our lab, has been generating reports on a weekly basis. And then that information is, again, disseminated across the country. So at the beginning of this, and until quite recently, this whole system was done on a weekly basis. So on a Friday, they call it Metadata Friday, they would upload all of the sequences that had been generated that week, as well as metadata. And now it's gone to twice weekly. And soon moving to daily, but we're not quite there yet. But with civet, in the intervening periods, you can pull out your sequences, but also add sequences into the tree if you're interested. So, for instance, if you've got a nanopore machine, you've run your pipeline, it's right hot off the sequencer, you can get information about your genomes immediately and feed that straight back. The genomes don't have to go anywhere. They can be fed straight back into public health services. In terms of the report that's been generated, we've tried to put an emphasis on what the data can and can't tell you. In terms of comparing two genomes, if you have two very different lineages, you can rule out transmission. If they're present in like two local trees, you know that there are a number of mutations apart and you can rule out transmission. If you have two identical sequences, you can't say anything. And this figure here is one that I sort of knocked up a few weeks ago, and it just shows on a single polytomy, so where sequences are identical, it checks the sample date from those sequences and shows how many, what's the number of, the length of time that on a single polytomy there can be two sequences that are identical but far away in time. Yeah, so here on the x-axis we can see the time since earliest sample on the polytomy. So you can see there's samples up in the 90s here. Yes, most of them are here, and when you actually take the median, it's 14 days, which fits really well with our estimations of the mutation rate of the virus. But yeah, so you can see that there are these outliers, and you can have sequences that are identical, but really, really far apart in time and space. This is an example report. So this was a real outbreak investigation that we ran. So I'm at the University of Edinburgh, but we're working with the Royal Infirmary down the road. And there was an outbreak in a particular ward in the hospital that I had been involved with. This was when we were developing civet, and we've been working with clinical scientists to try and see what information we want out of it. And so all of these, there's 1, 2, 3, 4, 5, 6, 7 query sequences. So each sequence corresponds to a different individual in the hospital, some of them were healthcare workers, some of them were patients. The patients were all in the same ward, and the investigation was sort of ongoing. So this is actually the third iteration of this report, as more sequences were generated and more patients joined the ward and got infected. So, but what this investigation actually showed was that there were two distinct lineages. So rather than a single ward being a single introduction event, this outbreak, it was actually two separate outbreaks and two separate, at least two separate introduction events, because of course if the sequences are identical, that doesn't really tell us a huge amount. And actually, as the outbreak was ongoing, I reran the report, and there's an updated report where we had extra sequences. Again, it's just two separate clusters of diversity, but that sort of information is useful. And so we're sharing things like we've got our own UK lineages defined, but we also assign them a global lineage and a phylotype, which is again just codifying the tree. Civet is quite flexible, so the code is all up online, but you can color by and display custom fields. So you can color by things like healthcare worker status, whether they're present in a care home or not, and different levels of geographical information. And recently Civet had a major speed up by re-implementing some of the tree parsing algorithms in Java, which is the same for llama as well. Okay, so that's me finishing up now. I think I'm okay with the time. I wanted to just thank everybody involved with the Lineages project. So the people named here are those specifically involved with the Lineages project and Pangolin and llama. And thank Rob for hosting his tree online, and thank Anthony and David for helping develop the pangoLEARN interface as well. So also we now have a website, which I spent a lot of evenings on front of the TV making. So if you want to check out more information about lineages, we've got a page for each lineage, and it gives you some links to resources, how to use Pangolin and llama, and how to suggest new lineages as well. So yeah, that's me. I don't know if you've got questions, but thanks very much for listening.
Well, thank you so much. That was a fantastic talk. You guys have been busy over there, doing a lot of stuff, a lot of work. I really appreciate you taking the time to do this. I will throw it out to the group to see if anybody has any questions, and if not, then I'll start in because I've got a few myself. So anybody have any questions? Oh. All right. Well, if not, then I guess I will get started with mine. So I know you said in the beginning that you had worked with Emma Hutcraft. And so I know that she is a part of the kind of next strain team. And so I was wondering, yeah, what kind of differences or similarities are you guys seeing between the kind of like classifications that they have kind of come up with between, you know, the next strain kind of builds of things versus how Pangolin is assigning these things? And is there a significant difference that you're finding between the two and how they're doing these assignments, and does it really affect how you're viewing the data?
So I think yeah. So me and Emma worked together on that figure. There's a paper that is coming out in. We'll see. It's in press at the moment. Not in press. What's the one? In review, and we've got reviews back. But it's just an overview of lineages and clades going around Europe. So that was where that figure's from. So I think the main thing is that lineages and clades are both suited for quite different purposes. So if you're interested in looking at sort of like changes in SNP profiles over time, and actually even something like the DUG, the D614G mutation, if you're looking at that and want to see how it is going to be changing over long periods of time, I think clades are probably quite useful. But if you're trying to do outbreak investigations and you're interested in introduction events on a finer scale, I think lineages, because they've got this hierarchy and because they've got the finer scale information, that is probably the more useful piece of information, I think. I think they've got different applications. They don't conflict in any way. You know, NextGen have got their system, and it looks very nice and clean on the tree. So I think lineages are they're not as well displayed, but I do think that for querying and trying to compare different introductions, and particularly in the UK, so we've got a huge amount of sequences from the UK. I think at the moment it's like 35,000, 37,000, something like that. And because of the lockdown, we're seeing a lot of geographical clustering. And now that things are starting to open up, actually using lineages, we're able to tell these things apart quite easily. Yeah, so I don't know if that answers your question. Yeah.
Yeah, no, I think it does. And I think that it kind of leads me then into the, like, how wide of a kind of variety of lineages are you seeing in the UK?
So a huge amount. The thing about the UK is we've got a separate level of information beyond lineages as well, because sort of in terms of introduction into a new geographic area with the UK lineages, we are going beyond the country level, whereas because I'm here, so we've got UK lineages as well, which are sort of smaller in scale than global lineages, and yeah, in terms of introductions into the UK, we've definitely seen that since lockdown began, it's sort of local transmission events rather than new introductions as a whole. But yeah, that's probably going to be changing fairly soon. Yeah, sadly.
Yeah.
So I suppose on a very local scale, if we're talking about within a hospital and say for that specific outbreak case, it's useful information to know whether you've got spread within the hospital or whether it's likely that this could all just be individual community introductions by visitors coming into the hospital. We've got research going on at the moment into care homes, which are like a huge source of infection and spread across Edinburgh, particularly, and looking at metadata about whether it's healthcare workers, whether there's like a shared lineage between certain health care homes, whether it's actually more localized, or whether there's particular lineages that are present only in care homes, or whether it's very clear that it's all just the same as in the community. So we have stats on, and civet, civet can run very specific analysis to summarize what is locally spreading within the community and compare that to the particular outbreak that you're investigating as well. So so on a very local scale, I think there is real information that you can use to inform like clinicians, to inform decisions in the hospital and the infection prevention control teams. On a sort of more zoomed out level than that, on a weekly basis with COG, we're reporting that information back. So we're reporting summaries, but also the fact that now all of this data is centralized and it's accessible to anybody who wants to run civet. And Public Health England is running civet. So if they have an outbreak, not necessarily just in a hospital, if they're looking at the community, they now have access to all of the lineages that are circulating around the UK, and they're able to compare, see what the source is. Recently there was an outbreak, and it wasn't, so we've got Scotland and England, and the outbreak was, it seemed to be sort of cross-border, but we weren't sure. There was like an outbreak on both sides of the border. And recently civet was used to confirm that actually it was one large outbreak rather than multiple small ones. And especially now, I think it will be important to know whether these flare-ups, because we've managed to dampen down a little bit, but we've still got quite a lot of circulation, but it'd be important to know whether they're introductions from abroad or whether they're from movements within the country and they've been imported. So I think being able to tell apart these different lineages is quite useful. And with the intensity of sequencing that's been going on, we can do that. So. Yeah. But everything that we've got up and running on the bioinformatics side is all open source. So if you want to look at the code, you're welcome to use it and adapt it. And particularly with llama, what we have with civet is a very specific case dependent on the UK. But llama is sort of, I wrote it as more of a boilerplate. People could take this. Yes, it can be used to identify new lineages. And actually, for the US, if you are seeing, say, that the next strain clades are just sort of everywhere, maybe with more local lineages, you guys can actually feed that information back into the lineage system so that the smaller lineages could be just getting assigned automatically with Pangolin as well. Yeah, so feel free to take llama, adapt it if you want to use it, if you have access to some genomes.
So I have another question, and this kind of has to do, I guess, a little bit more with like the infrastructure of sequencing in the UK. So do you know approximately what percentage of the positive samples you guys are actually sequencing? And so like we can get kind of an idea of that.
Yeah, so it's varied across the outbreak, right? So at the beginning we were sequencing, in Edinburgh specifically, we were sequencing almost everything. We got a GridION quite early on, and so we've had sequencing capacity here from the beginning. But I think the sampling, so the funding for COG has come from the government, and 50% of the funding is supposed to go towards surveillance, and 50% is supposed to go towards outbreak investigations. So a lot of the sequencing centers, just because of the way we're set up here, are actually based in hospitals. So a lot of the sort of, so 50% were going into just, and honestly, because there were invested interests, more were going into outbreak investigations than the community surveillance, and also just access to samples and things like that as well. So it's changed throughout the this last few months. Currently, we're again sequencing, I would say, nearly every case that comes up. But in the centre, when we were at our peak, it was much lower proportion-wise. I think in terms of percentages, yeah, no, I couldn't tell you. But we've got, if I divided the number of cases with the number of, sequences we've generated, it's probably still quite low. But it feels like a huge amount of data to me. And especially now that our focus has moved and is looking at these flare-ups and outbreaks. It's more sort of specialized sequencing that's going on. So you might sequence an entire, 30 sequences from a particular outbreak because you want to know how many introductions has caused that outbreak and stuff like that. So.
So I know that we've got a minute left before the time is set to expire. I am happy to stay on a little bit longer if you have time, Anya, and then if anybody else wants to stay on a little bit longer too. I've got maybe a few more questions, but I just wanted to be cognizant of everybody's time and let you know. And I don't know, Sean, hopefully we have the Zoom account for a little bit longer. Okay, perfect. That sounds great. Thank you so much. All right, so for those of you who have to go, thank you so much for